Next, I'm going to talk about experience with Oncomine Myeloid research assay. This is the disclaimer. Why choose Oncomine Myeloid NGS assay? There are a couple of reasons. Firstly, it's the affordable platform with iron-tolerant S5+ and Chef. Secondly, there are 74 genes with relevant mutations and fusions. And also, another reason is the small sample size. We can use 10 nanogram of DNA and RNA from blood or bone marrow. There is another reason is the single workflow. We process DNA, RNA sequencing simultaneously with automation for library prep and templating. And another reason is the shorter turnaround time. The workflow is for 3 days. We basically run 3 to 5 times per week to meet that turnaround time. For reporting, we use Oncomine Knowledge Base Reporter. This is the NGS Oncomine assay content. There are 23 genes with hotspots, 17 genes with 4 gene coverage, 29 fusion drivers, and 5 expression genes. We slice 3 panels for the testing. We have AML, 56 genes, MPN 5 genes, Heme 74 genes. The most challenging part is the FLT3 ITD region. So this region, challenging targets are very well covered, and there is also a dedicated software to annotate FLT3 ITD. Another challenging targets are well covered is the Cbpa 4 gene coverage. You can see it is pretty well covered. Those black bars showed all the coverage. For fusion partners, there are many known partners. For example, for KMT2A, there are 199 partners. For BRAF, 95. For ETV6, 52. ABL1 is 49, and the RARA is 45. What about NGS validation guidelines? So we follow Jenny's paper in 2017 for sample requirements and for validation. So the minimum requirement is 59 samples. It's harder to understand the formula to get a number, but that's what it is required. So we actually did 76 samples for validation. And there is another guideline from CAP required for NGS validation, especially to meet the requirement for all the molecular pathology checklists. Next is the standards and the guidelines for the interpretation and reporting of sequence variants in cancer. So this is the Marilyn Lee's paper in 2017. It is recommended to include the tier information into the classification and the reporting. Next, I'm going to talk about assay performance characteristics. I'm going to mention about the accuracy, precision, analytical sensitivity, and specificity, and the lower limit of detection. This is the accuracy. So on the table on the top of the slide, I showed all the variants we have validated. Among 109 variants, there is only one of them did not identified by our assay. I'm going to talk about this in the later slide. So the accuracy is 99.05%. If you look at the lower part of this figure, the x-axis shows all the variants. On the y-axis is allele frequency. The blue bar shows all the allele frequencies from different lab. We got samples from them. The orange bar shows the results from our lab. You can see for most of these, the allele frequencies are very concordant. For precision, we did two assays. One is the intra-run assay. The ABC stands for triplicates in the same run. Also, in the figure, you can see on the x-axis, they are the variants. The y-axis are the allele frequencies. ABC stands for different color of the bars. They are very concordant. The second part for the precision is the inter-run assay. The tests were run in day 1, day 2, day 3 by 2 techs. You can see the results for day 1, day 2, day 3. The allele frequencies are very consistent. For limit of detection, we ran a sample include the FLT3 ITD region, and we did a series dilution from 12% to 0.75 with expected DNA mutation level, and the allele frequency by SQL is on the right side of the slide, and you can see the allele frequency is very concordant with the expected level. The cutoff is 2.5 for FLT3 ITD. The mutations detected from Seracare reference DNA, you can see from the list of the genes and the variants and also expected variant allele frequency is in the middle of this table. And on the right side of the table, the allele frequency is obtained from our assay. Those expected number are very concordant with expected allele frequency. The coverage are pretty high. It's usually around 2,000. The fusions detected from Seracare reference RNA, we identified all 9 transcripts from the sample. The read count is above like 10,000 for most of these. Next is the data from the fusion we tested for patient sample. You can see we obtained most of the sample identified by RT-PCR or FISH or NGS from a different lab that was on the second column of the table. The fusion detected by SQL is on the third column. We detect all of the fusion by NGS except for one, which is highlighted in red. It is PDGFRB, identified by PCR from another lab, but not detected by NGS from our lab. So we repeated like 2 or 3 times, then we finally figured out the primer for the RT-PCR and the primer for the NGS are different. Summary of clinical research data. So far, we have run 1,367 samples. 34% are from AML patients, 10% from MDS patients, 3% from MPN, 53% from other. The abnormality rate is 56%, including mutations and fusions. Next, I'm going to show you the molecular profile for myeloid malignancies by NGS in terms of the mutations. On the x-axis, these are all the genes, and the y-axis stands for the number of patients with mutations. So the highlighted area for the top one mutation is from TET2. You can see most of these mutations are from MDS patients and also from the AML patient, very rarely from the MPN patient. For the JAK2, the next arrowed one is mostly from MPN, some of them from AML or MDS. For FLT3, of course, it is more from the AML patient. For TP53, it's about half from AML, half from the MDS. What about the fusion profile? We have mostly CBFB MYH11 translocation. Next is the RUNX1, RUNXT1, and then BCR-ABL1, KMT2A rearrangement, and PM-ARARA. For the potential assessment of prognosis indication for AML patients in clinical research study, we know that by cytogenetics there are 3 big categories for risk stratification: favorable, intermediate, and poor. So for co-binding factor inverted 16 or the translocation 821 or translocation 1517, they are favorable prognosis. However, if you have a molecular abnormality with the Kit mutation, the prognosis information will be changed. I'm going to show you some examples in the next slide. This is the co-binding factor AML with Kit mutation. Totally, we have 34 patient samples with co-binding factor CBFB rearrangement. 12 samples has RUNX1, RUNXT1 translocation. 6 of the 34 samples had, if you look at the table on the bottom, CBFB translocations in total, we have 34 patients. 6 of them had a Kit mutation, and 4 of these are on exon 17. And for RUNX1T1 translocations with Kit mutations, there are 5 samples, and all of them are from exon 17. So if the samples patient has CBFB MYH11 translocation, also a Kit mutation on the exon 17, it is not really bad. If you see the line, the red color, and compared to the wild type Kit mutation, it is not really bad. It's considered still good. The next one is the RUNX1, RUNXT1 translocation, and also coincide with Kit exon 17 mutation. It will become the poor prognostic factor. You can see that red line with Kit exon 17 mutation compared with the blue line, the Kit wild type, the survival rate is worse. In summary, NGS has become the standard for molecular assays and represents huge potential value to oncology clinical research. Guidelines for comprehensive biomarker analysis in hematologic disorders are available. Oncomine Myeloid research assay provides a more comprehensive genetic profile of hematologic malignancies, samples, and relevant information. Oncomine NGS assay were chosen because of low sample input, improved turnaround time, intelligent NGS content design, single workflow. In the future, NGS results have the potential to change the paradigm of cancer therapy and facilitate precision medicine. With that, I thank you for your attention, and I'm ready for the questions.